All right, this is going to be the intro to photosynthesis and a little bit about pigments. So you guys remember that we have been doing primarily chemoorganotroph catabolism, right? Where we start in prototypic chemoorganotroph catabolism with glucose, and then it goes through some sort of glycolysis, then pyruvate oxidation, and then the Krebs cycle. And the job of these three steps is complete oxidation of the organic carbon, giving us electrons in mostly NADH and FADH2, and then energy in ATP, and like the GTPs that are made in the Krebs cycle. Okay. Then in the electron transport chain, and in this case oxidative phosphorylation, the electrons are used to indirectly create more energy as ATP, okay? And then the chemoorganotroph, can you guys see that? You cannot. The chemoorganotroph cell uses this ATP to power all different sorts of things in the cell, and also uses the PMF created by the electron transport chain for a few things in addition. And so this is the general process of chemically based catabolism. Okay, so in photosynthesis, it's a little bit like this backwards, okay? So photosynthesis, I really want to use a different color, but I do not have one. Okay, so sorry about that. So photosynthesis uses, so energy is always like the squiggly line arrow. I do not know who decided that, but it is true. So energy from the light, so if you are, well, really any sort of photosynthetic organism, this mostly means the sun, although you can grow like your plants inside and stuff, but so the light comes and it gives some energy, and then that energy is captured and used to make ATP and the electron carrier NADPH, right? And so then, and you guys will remember that NADPH carries electrons for anabolic or building reactions. And so then the ATP and the NADPH are used to build glucose. Woohoo! And so photosynthesis is, very generally speaking, backwards the chemoorganotroph catabolism. And so there are actually two kind of different sections to photosynthesis. We'll see if you guys can see this on here. So this bit, where energy from the light is harvested and is used to produce ATP and NADPH, is called the light dependent. Just pretend like you can see that. I can zoom in some. This part is called the light dependent reactions, or the light dependent process. This part, where we use the ATP and the NADPH to make glucose, does not require light because we've already made the ATP and the NADPH. So this bit is called the light independent process. And so we are going to learn about both of these. This light independent process primarily happens in the Calvin cycle, not to be confused with the Krebs cycle. No. So this is a little bit stupid, but this is how I remember it. So this guy's name, I kid you not, was Melvin Calvin. So you can think to yourself, you're unlikely to forget Melvin, right? Like none of you will forget Melvin. So just think to yourself, Melvin Calvin, like that sounds snappy. You can imagine him, he has a bow tie, suspenders. Melvin Krebs, like what? No. So Calvin cycle goes with Melvin and it builds things because Melvin makes stuff, right? So there you go. You're all fixed up. Okay, so this is our just to get you going before we start on the actual slides. Okay, so this is your official photosynthesis introduction slide, where your light-dependent reactions are the ones that require the sunlight. So somewhat obviously, these happen primarily during the day. In super old textbooks, like I don't know how you guys learned this in Gen Bio, sometimes these are called the light reactions, and the light-independent ones are called the dark reactions. People have quit doing that because calling this light independent step the dark reactions made people think that it only happened at night, when in fact it happens all the time, whether there's light or no. It is light independent. I wish that you guys could see me because I'm doing all these fabulous hand motions and you're just completely missing them. So the light dependent reactions, again, capture light energy, which is always drawn as this squiggly arrow thing, light energy. Actually, this is a radiant energy, but for our purposes, it's light. So light energy is used, and then we actually are going to get electrons, well, you'll see, largely from water. And then we will make, in the short run, a PMF, and then that PMF will be used to make ATP, and then the electrons will go to make NADPH. And so all of these things are being produced to then support the actual building process, which is going to happen in the light independent reactions. And so we are going to, in the light independent, but the cell is going to take carbon dioxide and some things that we'll learn later and make it into glucose. So carbon dioxide, bizarre but true, if you did not know this, is considered to be inorganic because it has a carbon, but it's not considered an organic compound. And so if you hear of a process that fixes, this means that it turns the compound into a biologically usable organic compound. And so fixed carbon would be like pyruvate or glucose or something organic that the cell can continue to build with. CO2 is not considered to be an organic compound. Okay, so hopefully you're good on that. So how is it that the sun is magically captured and turned into PMF and then ATP, and why can we not do this? So let me zoom out for you here. So this magic happens by pigments, and we cannot do this because, well, we're not really pigmented. So pigments are how light energy is captured for use. This chlorophyll, which is, of course, the primary pigment in land plants and green algae, and bacteriochlorophyll, which is the primary pigment in some photosynthetic bacteria. Yeah, we'll get to that on the next page. Look like this. They are a magnesium-containing porphyrin. And what that means is they are this planar ring structure with a magnesium in the middle. And so if you have ever heard Dr. Bradshaw or Dr. Hayes talk about their research in photodynamic therapy, where you give the person the drug localizes to the cancerous tissue, and then you shine a light, and the drug will preferentially absorb the light and then spread the energy to the cancer cells, ideally destroying the DNA. So that is exactly, what happens here is this multi-ring structure will work to absorb light energy, and then that light energy is passed to an electron to give it energy, and then it will go down the electron transport chain after that. This is well, we'll skip that. It's on the next slide. Okay, so primary pigments. Do you guys remember when we talked about primary and secondary metabolites and how primary metabolites were the ones that were required to be alive? If you are a cell, you do this metabolic process and you need these primary metabolites. But secondary metabolites are really helpful and could give you different advantages. This is kind of like that. So primary pigments are chlorophyll and bacteria chlorophyll. And if you are photosynthetic, you must have one of these two pigments. These look vaguely greenish. So chlorophyll is found in oxygenic phototrophs. And so phototrophs are things that get their energy from light. And of course, if they are oxygenic, and this is right, genesis means making, and so oxygenic phototrophs make oxygen. So you will be unsurprised to learn, well, you might be, but you should not be surprised to learn that this happens when water is split. The electron is actually donated to the photosynthetic electron transport chain, and then the oxygen is secreted, for lack of a better term, from the cell. So oxygenic phototrophs are photosynthetic organisms that produce oxygen. These include plants, as you know, and very importantly, cyanobacteria. And so we will mostly learn this photosynthetic process because it is super well studied and should be the same as the one that you learned in Gen Bio 2. We will still cover anoxygenic phototrophs, which somewhat obviously right there, the equivalent of anaerobic bacteria. And so the initial electron does not come from water, and so they do not produce oxygen. That's just it. And so this little chart thing here shows you the Roy G. Biv visible light spectrum, and shows you both of these, the chlorophyll and the bacterial chlorophyll, have two absorption peaks. Can you guys see this? Let me zoom out a little bit. So chlorophyll absorbs in the red and the blue, and bacterial chlorophyll absorbs in the slightly more red and in the purple. And so the fact that they neither one absorb in this green bit, right? So the light that is not absorbed is the light that is reflected back to our eyes. And so this is why most/many photosynthetic organisms look green. The reason that they don't all look green is because of their accessory pigments, which we will get to now. So accessory pigments in our primary and secondary metabolite example are more like secondary metabolites. And so accessory pigments will absorb light that the primary pigment misses. And the more light the cell can absorb, the more energy it can produce. So we are all about absorbing more light. Also, the less random light energy is wandering around to damage the DNA, which we do not want. So go light absorption. So more absorption is better. And so this is chlorophyll, which again is in cyanobacteria and land plants. You will need to remember this. So cyanobacteria and land plants, it's land plants versus like kelps, which are not really plants, right? In case you've forgotten, kelps are giant protists. Nevertheless, here we go. So chlorophyll, which we will abbreviate CHL because who wants to spell out chlorophyll, is found in cyanobacteria and land plants. And if you might remember from the previous slide, chlorophyll is used in oxygenic photosynthesis. So both of these guys produce oxygen, which hopefully you remember from like 2 minutes ago, means that they get electrons from water. Okay. I don't know if you can see. We're just going to hope that you can. All right. So this is our chlorophyll absorption spectrum. Remember, chlorophyll absorbs in the red and the blue, and so that is why like many plants look green. So if a plant has carotenoids, which absorb into the like bluish green and the green, then this plant or this cyanobacterium can absorb more light energy, allowing it to make more ATP in the long run more easily. And so this cell would be more likely to look green or yellow or like maybe a little bit orange, like some of these attractive produce items here. And so, yeah, so accessory pigments are pigments that do a supplemental light absorption role. And the reason that this is really important is that the more energy the cell can harvest, the more ATP it can make. So the reason that if you like sit outside, you know, people like put on sunscreen so that you don't get cancer. Sunlight is incredibly energy containing. I know that sounds stupid, but just pretend like it doesn't. So sunlight has lots and lots of energy. And so when it shines on the cell, it can energize electrons out of their bonds, and then you have a free radical. So this is a big problem for the cell. So not only does the cell want to intentionally collect this light energy so that it can save it, the cell also would like to absorb this energy to protect itself from sun damage. And accessory pigments do both of these because they will absorb in the middle of the spectrum here, where the chlorophyll or the bacterial chlorophyll is not absorbing. This is a picture of my antioxidant from the superoxide dismutase set of slides, because by having this protective light absorption role, accessory pigments will function as antioxidants, and they're giant molecules that have a lot of electrons that are shared among the molecule, and it's that ability that lets them absorb light energy and kind of buffer against electron loss. So specific examples of accessory pigments are the carotenoids and the phycobilins. So the carotenoids are, I don't know if you can see them on here, but the carotenoids absorb the very end of the green, the blue-green, and then a little bit into the dark blue. And they are throughout phototrophs. So anything that gets its energy from light will have a carotenoid. Famous, well, not will, but may have a carotenoid. Famous carotenoids, I don't know if you guys can see this. It's like too much light. Well, I'm just going to read it to you. Famous carotenoids include lycopene, which is the red pigment in, seriously, you can't see that either? Okay, lycopene, which is the red pigment in tomatoes, alpha and beta carotene, which is in your orange vegetables, right? So like butternut squash or carrots or sweet potatoes or anything like that. And then this is lutein and zeaxanthin. So depending on your plant knowledge, you might know that ZEA is corn. So lutein and zeaxanthin are found in large quantities in corn. And so you will also find these different carotenoid pigments in all different kinds of plant foods, including like kale, spinach. They're just not present in as high of quantities. They're still there, and you can still, like, this is part of why kale is really good for you. But if you can, if the food product is that color, then they have a super high amount of that carotenoid pigment. The other type of pigment that we will refer back to are phycobilins. There are all different kinds of phycobilins, including phycoerythrins, which, so remember, the color that we can see is opposite of the color that is absorbed, right? And so erythrocytes are red blood cells, and so phycoerythrins, if you get them by themselves, look red to us. And so that means that they will absorb light primarily in the green area, and phycocyanins look blue to us, cyan, and so they will absorb light primarily in the yellow and orange wavelength, because then they will look the reflected color and not the absorbed color. In any case, there's a whole different range of phycobilins. They will absorb, in red, orange, and green wavelengths, and they are found only in cyanobacteria, whereas carotenoids are in cyanobacteria, plants, and anoxygenic photosynthetic bacteria, because you will remember that cyanobacteria are oxygenic. They produce oxygen. All right, we have just 2 more on this. So here is the way that this works. It is the niftiest thing ever. All right, so you guys might have seen like solar panel arrays. This is that, except it's in the plant / cyanobacterium. And so you have got, this is an actual electron micrograph from inside of a cell. So you've got your, right in the middle is what is called the reaction center. And the reaction center is actually 2 molecules of the primary pigment, right? And so the primary pigment is going to be the chlorophyll or the bacteria chlorophyll. So 2 molecules of that are called the reaction center in the middle of this photosynthetic array. You can't see that at all. Okay, they're right here in the middle of the photosynthetic array. Then, in rings around that are what are called either the antenna pigments, well, you can read here, antenna pigments, also called the light harvesting pigments. And so light harvesting pigments are pigments that harvest energy from the light. So these can be more molecules of the primary pigments, which are chlorophyll and bacteria chlorophyll, or these can be the accessory pigments, the carotenoids and the phycobilins. And so antennas are, or also called light harvesting, if you see LH either way, will be in rings around the reaction center. And so what happens is the light energy is, remember the squiggly arrow always equals radiant energy. And so the light energy is absorbed from the sun into each of these different antenna pigment molecules, and then that energy is transferred, bink, 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 down to the reaction center. And so it is not incumbent on the reaction center to absorb all of the light energy. This is what makes this awesome. So this humongous, comparatively speaking to the size of the cell, this humongous array of photosynthetic pigments, both primary pigments, which are chlorophyll and bacteria chlorophyll, and accessory pigments, which absorb at different wavelengths, different than the primary pigment, not necessarily different than each other. So these primary pigments and these accessory or light harvesting pigments will form in rings around the reaction center, which is a pair of molecules of the primary pigment. So all of this light energy is absorbed, and then it is all passed right there. And then, this is the coolest part, the energy comes from the reaction center chlorophyll to the electron. And so this is just shows the electron like jumping over here. But here is what I want you guys to think of. So here is our lowly electron, right down here with oxygen at the bottom of the reduction potential chart. So the light energy comes down, and the electron's like, woohoo! And it pretty much jumps all the way up to the top of the electron potential chart. Like it doesn't, because we're not adding it to glucose right then. But energetically speaking, it does. And so when the light is absorbed, all of this light energy is given to this electron, which gets a lot of energy, and then it is going to go through an electron transport chain. And then, number one, where are electron transport chains located? In the membrane. Which membrane is the only one in bacteria? Right, the cytoplasmic membrane. And then, as the electron goes down the electron transport chain, a PMF will be generated, and just like in other electron transport chains, this PMF will be used to make ATP by the plant or the cyanobacterium. So this is, we're going to get to this again, but this is the cool pigment part. So because the pigments are collecting light energy to power, oopsie, this like zoom in knob is backwards, I'm telling you. Because the pigments are collecting light energy to power electron transport, and electron transport must always be in a membrane, the pigments are located in the membranes. So there's the plasma membrane, which we already talked about. Then there's little fold-ins of the plasma membrane. So if you've got your cell and then you like this type deal. And the reason that you would want this is if your pigments are in the membrane and the only way you make energy is by light, you need as much surface area for those pigments as possible. And so you can get these fold-in membranes. If it's just a little bit, it's called an invagination. If it is a lot, you can see these giant fold-in membranes, then it is called a thylakoid membrane. And if you will remember from Gen Bio 2, thylakoids are the membrane sacs inside of the chloroplast where photosynthesis happens, where the light-dependent part of photosynthesis happens. And so this is one of the structural similarities between cyanobacteria and chloroplasts that supports the endosymbiotic hypothesis. If you guys remember when we talked about that for exam 1. The final place that you can find pigments is something called chlorosomes. And so chlorosomes are this. Chlorosomes are basically this big stack of pigment molecules that are all feeding into this reaction center, which again, the reaction center is a pair of molecules of the primary pigment. And so the chlorosome is this, I'm reluctant to say bundle because, well, it's more of a collection. A chlorosome is a collection of photosynthetic pigments that will all feed down to a single reaction center. You can see them here, these oval, can you see those? Let me zoom in some. So these oval things that are all the way around the inside of this bacterium, here's a bunch of them right here, are the chlorosomes. Sometimes there is a membrane around them, sometimes there is not. Sometimes there are proteins, sometimes there are not. But chlorosomes are these oval or rectangular collections of pigments immediately under the membrane in some bacteria. And again, those will absorb the light energy, pass it to the reaction center, which is a pair of molecules of the primary pigment. And the primary pigment, the one that is absolutely required for photosynthesis, is chlorophyll or bacteria chlorophyll.